confidence in your observations as far as life detection goes. So I'm going to throw a lot of techniques at you. Here's the surface in reflected light. This is transmitted light at the surface. You can see this small feature here that slowly comes into focus about 5 microns in. And these little blebs here are all 1-2 microns, round about bacterial size. When I do the Raman on them, they're absolutely filled with this reduced carbon signature that's been used and touted as a definitive biosignature for early life on Earth. It isn't. It's a definitive signature of reduced carbon with a lot of aromatic functionality. So when I took that and did a FIP section across that feature, the thing we pulled out was this. This is around about 100 nanometer scale bar. This is calcium sulfate. This is an iron sulfide, a pyrrhotite grain, magnetite, and it's surrounded in a rim of clay, montmorillonite. Something's happened here. There's a lot of volatile degassing. We think that was due to impact. But this structure is extremely complicated in the submicron realm. A lot of mineralogy going on there. So I use soft transmission X-ray microscopy to have a look at it, and this gives me the functionality of carbon in this system. Here's an iron map. There is our calcium sulfate grain. Right, this is all iron, and you can see that the reduced carbon from mapping this peak here all occurs around about where the magnetite and pyrrhotite was. If you look at that carbon, it's dominated by aromatics, but there's a lot of CO functionality, single bond, C double bond, carboxylate. There's a little bit of nitrogen functionality in here as well, CN double bond C, nitrogen heterocyclics. So it's something that contains a lot of organic functionality as well as aromatic. Now this will give me my D and G carbon in the Raman. The rest of this, no, I can't see that, unfortunately. So is it Martian? Well, the answer is if you use the nanosims and punch down. All of my observations, I tried to make underneath the surface of the thin sections I look at, or underneath the surface of fresh fracture surfaces, to get away from the haunting spectre of terrestrial contamination. So this is a magnetite map and a reduced carbon map of this feature here. And if I do the D2H from this with Eric, what we find is D2H around about 3,000 per mil. Big error bars on this, I know, but the D2H at the top of this section, which was contaminated, is minus 50, to give you some idea of the contaminant ratio in this. So this is all crustal Martian signal, all up here. Here's Earth and terrestrial water down here. Our polymer is down here, and our signals are up here. So this is some form of Mars crustal water interacting with the sample to produce these organics, maybe. So that's where our analysis lay for a little while. Then I sent this off for time-of-flight sims. Time-of-flight sims is, you blast an ion beam at something, you click go on a timer, and you time when the ions reach. So hydrogen will reach fairly quickly. Larger molecules take longer, and it's extremely sensitive. You can get down to like 1,000th AMU resolution on a good day with a following breeze. So in this sample here, we can see a lot of chlorine, some sulfur, and I want you to just look at this spot here. Sulfate, same as we had. CN, yes, CNO. We didn't have that in our Stixum data, but we did have this. And there seems to be some carbon-sulfur bonds, iron oxides, iron chlorides, maybe some perchlorate. If I show you the expanded maps, a lot of nitrogen functionality here. So we've got chlorine, sulfur, nitrogen, carbon reactions going on in this system. And you can see where CN and the sulfur signals are all, it's exactly the same sort of mineral assemblage that we looked at in the Stixum data. If I take this meteorite and heat it up to high temperatures and do a combustion at several temperature ranges, I can get the nitrogen isotopes out. And the nitrogen isotopes in the high temperature stuff that we're looking at is around about, on average about plus 40. This is normal terrestrial values. This is all terrestrial contamination. So in a series of observations, including some lab-based work, we looked at getting rid of organics contamination. We found that below 600 degrees C, it was all terrestrial contamination. Above 600 degrees C, we actually started to look at a Martian signal. And this tied very well with the literature. Monica Grady, Ian Wright have been saying this since the mid-80s. But they never had the provenance of the carbon they were looking at. They just labeled igneous carbon. So if I go above 600 degrees C and I run in this meteorite and I run GCMS on this, so this is these species are produced, you know, you get the gas, you put it down a GC column, it's split and then into a mass spec. These are all the species that are being released above 600, right? A lot of these species are really labeled. They should come off like 100, 200 degrees. They shouldn't be there, right? So they are there, including the aromatics. This is thiophene. I want you to remember that one. Here's our nitrogen heterocyclics. This is, again, a lot of aromatic functionality here. So what exactly are we looking at? So recently I've been doing a lot of FIB-TEM work in Germany. I'm going to look at these two meteorites. This here is a 3D map of both red magnetite, blue reduced carbon in this meteorite, NWA 1950. And you can see that the reduced carbon layers in between all these magnetite grains here. The same thing in NACLA, in mesostasis. And so when I fib those things out and have a look at them, you can see here, this magnetite grain here has like a comb-like texture here. This is all organics in here. And it's filled with silicon, aluminium, iron, a little bit of titanium from the EDX. But there are no lattice fringes in this. This is all purely amorphous. All in here. And again, you can see these laths. These laths are ex-solution lamellae. These are titanium rich, as you can see here. The titanium pore laths that contain iron. This is a titanomagnetite grain. You can see the iron is missing here, apart from where the titanium's coming through. And if you look at the carbon map, you can see some functionality here between the titanium laths. There's even nitrogen in this one. A lot of chlorine about. Chlorine is all the way through this area here. So and here's the oxygen map. There's a bit of oxygen going in here. This is a Raman map of actually the fib section itself to show off the resolution of my Raman. Is macromolecular carbon, and this is the titanomagnetite grain. This is rotated 90 degrees to the right. Looking at the other meteorites and looking at that massive magnetite that had all the reduced carbon in it. This is titanomagnetite, apatite, pyrrhotite, and you can see along these laths here, there's evidence of basically corrosion, ex-solution. You can't join those two things together. This is all missing, right? So we see a lot of evidence for these corrosion textures. This is the NACLA again, and the area that I was showing you on the elemental maps. And you can see that these laths, I've just increased the contrast. So you can see just how prevalent these laths are and how corroded between these laths is. So is there anything that explains that? I mean, it certainly doesn't look, life can be involved in microbial corrosion, right? Life can corrode metals. That was my PhD and postdoc. I got bacteria to decontaminate nuclear structures, stainless steel in nuclear structures. So yeah, organisms can do that, but by far the most common way of doing that is through a galvanic cell. You have two unequal metals in contact with a fluid that contains chlorine. If you get stainless steel, put an elastic band around it and drop it into some water, into a brine, seawater, you will cut that stainless steel in half with the elastic band over about 6 months. because the elastic band stops oxygen getting to the surface, so you set up a differential aeration cell, and that's enough to set up a galvanic couple between, even though it's the same metal, right? So that process is well known on Earth. So basically what's happening? Well, there's an entire industry now on the electrochemical reduction of CO2. And this is in an attempt to get CO2 out of our atmosphere and actually do something positive with it. As you know, it's positive to getting it out of the atmosphere, but you can also make a whole slew of organics from it. And it basically is an electrochemical cell. You're splitting water to hydrogen and OH in this system. And when you do that, you allow any aqueous CO2 to combine with the hydrogen to make, in this case, formate, CO, methane, a lot of hydrogen. So around that grain, if it's corroding, you're producing a lot of hydrogen, some OH, you've got chlorine there as well, and I'll go into that in a minute. You definitely have a weak bicarbonate solution in there, because we pick up, in NACLA, you pick up carbonates. But below pH 4, those carbonates aren't going to form. And in this system, if it drops the pH enough, it will actually start to reduce CO2 with the hydrogen evolved from the electrolysis reaction. Can magnetite do this? The answer is yes. It's known in the literature for a long time. But no one's looked on this kind of scale. Remember, those scale bars are in the nanometer realm. So this is the industry that is now growing around this. You basically use electrochemistry to take CO2 out of the atmosphere, make liquid fuels, put it back, and you start this loop here where you're at least, discharge neutral as far as your petroleum products goes, and you have basically a petroleum economy that's cleaning itself up as it goes along. That's the dream. I think we could all think of why that won't exactly happen. So these are the reactions. This is the voltages needed at various from the electrodes to make this happen. Now, in industrial systems, you have electrodes that are huge. The distance between them is massive, so they have to pump a lot of voltage in. In the micro realm, the difference between that titanium-rich lath and titanium-poor lath is angstroms. So the charge density will go through the roof, and you'll be a lot more capable of making these reactions in this system. And also, you've got all sorts of things like silicon and all that helping you with those reactions. In 1990, to show you how long this research has been going on, this paper, Electrochemical Reduction of Carbon Dioxide and Various Metal Electrodes in Low Temperature, i.e. 4C, aqueous carbonate media. And what you find is, depending on the metal that you choose, you like to produce hydrogen, hydrocarbons, CO, and formate. And the metals we have in here are basically iron, chromium, titanium, vanadium, bit of manganese. So all of these reactions and all of these products are able to be produced in this system. What's more, since 1990, iron has been shown to be producing some hydrocarbons and some formate in this system. Okay? So, since the Viking era, the Viking observations of Mars, there was a thing called the label release experiment, which is the only life detection experiments performed on Mars. They got a positive result according to the lead investigator. However, other observations on those missions showed that there wasn't enough organic chemistry around to explain these labeled release experiments. And the conclusion was that there was no life there, but it was an unknown oxidant. And it took until the Phoenix missions to find out that that oxidant was probably perchlorate. So this is perchlorate here. On Earth, I showed you there was perchlorate associated with these features in the TOF SIMS data. So on Earth, the way that you make perchlorate industrially is electrochemical treatment of brine, of seawater, and it'll get you these reactions that are about 1.2 volts. So this is a reaction that's extremely well known and constrained on Earth. So what I think is happening is hydrogen is being produced in that reaction, but also the oxygen, the OH, that's being produced is basically combining with chlorine in the system. What other evidence do I have for that? Well, this is a diagram of all the possible routes that methane could have been made. Now, since the MoMA and Villanova observations of methane in the Martian atmosphere, and they're followed up by the sample analysis at Mars measurements on Curiosity, people have been wondering where this methane comes from. Okay, and one source is obviously life. The other source is possible electrochemistry, just water-rock interactions. I've included my electrochemical thing in here because it will produce methane as well. And then there's methane clathrates have been speculated to exist here. So going back to that meteorite I showed you in 1950, where all the carbon is layered through. This is the carbon. This is actually thin section polymer. So I'm tracking where the contaminant is in the system before I fib it. And so when we fib a certain area of this, what you see here, this is a pyroxene grain, this is glass, this is all amorphous material in this crack. You zoom in, this looks very vesicular texture here, and it's all carbon. Sticksome data shows that it isn't carbonate. Not Sticksome, Eels data showed it wasn't carbonate. And as we zoom in a little further on that material, you can see it's very vesicular. And in certain places we have intact crystallites, there and there. And they're rimming, the crystals are rimming these vesicles here. Right? The inside is gone, but there's still rim of, like you can see here and here. And you can see that there's actually lattice fringes associated with here, but not in here as much or around. So what on earth are those crystals doing in the middle of macromolecular carbon I've been looking at, and what are they? Well, Eels mapping shows that the carbon, chlorine, and do contain oxygen. There's no decrease in the oxygen signal in this area. Right? So I have a carbon, chlorine, oxygen-containing phase. So we can actually look, that's a 5 nanometer scale bar, we can actually look at this and do an FFT of that and get the electron diffraction pattern. And believe it or not, there aren't that many things that contain carbon, oxygen, and chlorine and are crystals. One is phosgene, and the other two are clathrate-type structures. That is it. There is nothing else. Phosgene is bad news because that's otherwise known as mustard gas. It's deadly at 3 ppm. If you want to send humans to Mars, you better understand whether that's phosgene or not, because it's going to be a very bad day if it isn't. The type 2 and type 1 cubic clathrates, they could be anything. It's a carbon-chlorine containing clathrate system. That's all I can say at this point in time. The first clathrate discovered by Humphrey Davy in 1811 was chlorine, and it only decomposes at 8C. The chloromethane clathrate stability field is such that it'll form at Mars ambient at minus 97 Celsius, which is just another day or night on Mars, really. Also, it'll stay stable up to about 20C, which is roughly where we were with this meteorite. And who knows what the stabilizing influence of the macromolecular carbon around it was. So after I did the TEM, I brought it back here hoping to find a Raman spectra of it. So I put the Raman laser on it, gentle as I could, tried to get a spectra off it. But those crystals are 20 nanometers. I didn't stand much chance, and in the end I didn't get it at all. Just got a nice macromolecular spectra for what was around it. So going back to do the last TEM session, I thought I'll put it back in and see what happened, see what the Raman did to it. This is before Raman, this is after. It's completely reshaped itself. The carbon is reformed, the crystals are gone. So whatever it was definitely heat sensitive. And the carbon's evolved as well from here to here. It's shifted. So that whole assemblage is basically not that stable and very heat sensitive, which makes me think more and more that what we're looking at is not phosgene, which decomposes at minus 120 to a liquid, but actually probably a chlorocarbon clathrate. That's where I'm going to go from my meteorite studies to Mars Curiosity studies. I'm on an instrument called SAM, a Sample Analysis at Mars, which is a GCMS. It does a thing called evolved gas analysis, and it also has tunable laser spectrometer to get isotopes of both methane and CO2. We landed in a massive impact crater. It's around about 3.8 billion years old. It's 155 kilometers in diameter with a 5-kilometer uplift. And we're kind of scooting around down here, roughly. And during all that time, we've taken drill cores, analyzed them with an XRD, APXS. We have all the context data, but also we've been doing GCMS. One of the first things that we found, and Danny Glavin and Caroline Freyssinet have published on this, is chloromethane derivatives. Now, Viking found chloromethane and dichloromethane, and at the time they were dismissed as contaminants. However, during the SAM observations, almost every sample we've looked at contains a range of chloromethane compounds, right? So how are they there? Well, number one, it gives me a great deal more confidence that the clathrate I'm looking at is probably a chloromethane clathrate, given the stability of those things and the fact it's chlorine and carbon in a gaseous phase. And then all of these molecules, they all fit into a type 1 or type 2 clathrate cage. That's also known. But it also suggests something else. It suggests that that magnetite assemblage with all that carbon in it, that was fizzing, literally fizzing at that time. The magnetite is dissolving away, the titanomagnetite is staying intact. This electrolysis reaction is happening, and it's bubbling out. And that's what that texture was. These bubbles of chloromethane crystallizing out, the water's leaving the system, it's freezing rapidly. That sample was within 10 meters of the Martian surface, could have been minus 120 at the surface at the time. Water is leaving that system rapidly, and it's just locked it in. And it's hyperstable, no, not very stable, sorry. And these molecules are the molecules that were trapped in that reaction. Remember, we don't know how these were made on Mars. There's some discussion about that. But if I'm right with the electrochemical reaction, I can produce methane and chlorine gas right next to each other while I'm producing the perchlorate, right? It seems too coincidental, but more data needed. So this is two types of data produced by SAM. This is what's called evolved gas analysis. So as you heat, you monitor each M over Z. So this could be, say, M over Z 16 oxygen, and you can see it's released at this temperature here. And then we take a subset of this gas and put it through the GC and look to see what kind of organic signal it's giving us. It's been a bit of a journey because we do have a high background due to a chemical called MTBS-TFA, and if you want to know what that means, ask George, and he can also draw the structure for you. So when we look at SAM EGA, at Mojave, which is one of our samples, what we start to find is between 600 and 800 degrees, we start to find a range of organics appearing. So C1 to C5 alkyl hydrocarbons, single ring aromatic hydrocarbons, all appearing above 600 degrees C, exactly like my observations on the Martians, meteorites. Again, thiophene, I pointed that out in my Tessint data, dimethyl sulfide. Actually, yeah, I'll find that. Methane thiol, all these sulfur-based carbon chemistries that I showed you earlier from the meteorite work. This is another one called Cumberland. And again, the same, between 600 and 800, we're getting these releases. It's only tiny, it's low ppm. weak signals, but definitely there. They're there in almost every analysis. They're not there in our blanks. So they have a test bed, which is exactly the same as the instrument on Mars, and it's in a Mars chamber, and it's pumped down, and you can load samples into that. There are 2 more meteorites, NWA 7034 and DAG4 76, loaded last week. But this is the analysis that we did on Tisint a couple of years ago, 600, and we get the same refractory carbon coming out that we saw in previous. Same alkyl hydrocarbons, same aromatics, thiophene, same stuff that we've seen previously. And actually we pick up thiophene like molecules in the tough sims as well when we map in situ on that. So the summary of organics that we found at the moment, this is Tisint laboratory GC and in situ analysis. This is the Tisint testbed, which is similar to the SAM MSL observations, and this is the MSL observations. And still not seeing much in the way of carboxyl, but that's being debated because of the CO2 signal we get. No nitrogen at the moment, but everything else, yes. So looking at all the in situ stuff I'm doing on the meteorites, not only am I finding organics in that reproducibly, and I can explain them by abiotic reactions, but I'm also seeing the same speciation and the same sorts of organics being released on Mars with the Curiosity rover, right? So I have mutually confirming observations on 2 different planets, and hence my journeys to Mars more. Interesting for life is find the nitrogen. Everyone's been following water and now carbon. Carbon is one thing, but you can't make life out of carbon alone. It has to be functionalized. I've found a lot of nitrogen-containing species in my in situ data, but we haven't found them in the GC data on Mars yet. One of the problems about these things being habitable is the presence of nitrogen in the system. This is a terrestrial organism growing on Allen Hills 84001 that I alluded to earlier. This is a terrestrial actinomycete, and you have to be, these fibrils here are all joining together into a cord where the fibrils, fibers of the organism wrap together. This is extremely indicative in this species of nitrogen limitation, not enough nitrogen about. So this organism's actually telling me, yeah, there's stuff that I like to eat because it's heterotrophic. It's eating organics, so that it's actually indicating there's organics to be eaten. But those organics are really poor in nitrogen. And that's what we see from the observations. So summary of a SAM result. When I go through and look at all the SAM results we've got, there are actually a couple of results I can't explain. I can't falsify my null hypothesis that there's no life, which is really interesting, and that's a whole different talk. What are these processes? Are they important to Earth? So at this point in time, none of my observations tell me that there's life on Mars. I can falsify the hypothesis based on my meteorite work. There's 1 or 2 little things in SAM. It's interesting. But one of the reasons to go and look at Mars is to get n equals 2. One of the reasons to go is to find out where we come from. So years ago I did a bunch of analysis on mantle xenoliths, and I find the same magnetite reduced carbon association in inclusions in mantle rocks. Not sure how or where that's going to go at the moment, but it's there. It's not serpentine either. In Mars in 2020, a rover will actually go and start to drill and pick up samples. We tested these drill systems way back in 2011. Those samples coming back to the lab will help an awful lot to kind of look at those reactions in situ and try and ground truth the SAM results against the meteorite results. The great thing about doing sample return is we have the context of the samples. And meteorites, we have no context whatsoever as far as the general area that they came from. Is this electrochemical reduction process a universal universal process? Well, recently I've been looking at Enceladus and thinking about that, about these gazers that come flying out from Enceladus, mainly because Mars sample return is hindered by the fact that you have to go to a gravity well, get out a gravity well, go to a gravity well, get out a gravity well, go to a gravity well, right? The amount of fuel you need to get off Mars makes the mission a three-stage process. Rover to collect samples and cache them, rover to pick up the cache, take it to a rocket, blast it into orbit, and then an orbiter to come and pick it up and bring back. Enceladus is putting this out for free. It's just there. It's spewing out, it can be picked up. Just fly through it. But looking at the reactions associated with this, olivine water, so serpentinization keeps coming up. There's a problem with serpentinization. If you try and get it to happen in the lab, you either need a high hydrogen gas overpressure, or you have to use natural peridotites, not olivine alone. And the difference between the natural peridotite and the olivine is the magnetite. So I think that one of the problems that has hit this field for a while is the fact that it's the combination of olivine and magnetite that are causing, through this corrosion process, hydrogen to be produced and therefore methane coming out from serpentinized terrains. And if you look when they fly through these plumes, you've got a lot of methane, simple organics, carbon monoxide, carbon dioxide, well, they say complex organics. Complex to me is not 40 AMU. It's several hundred AMU. Also, they're talking about clathrate maybe in this system. All of it being spewed out into space for free if you can go and pick it up. Is it the same reaction that I'm looking at on Mars? And if it's the same reaction, well, if it is the same reaction, then Mars is actually telling us a lot more about our planet and how life formed here. This is a little out of context, but those hydrothermal vents are spewing out a lot of brine and therefore a lot of salt. This is a meteorite called Moynihan's and Zag. They contain salt actually within the meteorites themselves as part of the rock itself. And if you look inside those brine, which are purple due to radiation damage, you see these little vapor bubbles. And when we look at those vapor bubbles with Raman, this is with Mark Fries, you see reduced macromolecular reduced carbon here, but you also see chloromethane. You see the same exact organic chemistry going on in samples that are analogous to what's coming out of Enceladus, as we've seen on Mars. So at the moment where I am, and this is the arm wavy part, Martian macromolecular carbon formed from hydrothermal interactions and the electrochemical reduction of CO2. In that system, you will also reduce nitrogen to ammonia, so produce oxychloride, a lot of chlorine, and so that the reactions in that system of the carbon are based a lot on sulfur and chlorine chemistry, which is it's a bit concerning. However, George, in organic chemistry, chlorinating organics is one of the biggest ways of manufacturing new organic species, and the way in which these are produced, if chlorine is binding to, say, formate or those other organics that we see, then that may provide the pathway to the more complex organics that I'm seeing, because at the moment in experiment you can get to formate and then you're pretty much locked. However, I have a problem with those experiments. I won't go into that. Nitrogen-containing organics are formed by the same process, perchlorate manufactured by the same process. All of these disparate observations, methane, chloromethane, in the atmosphere, water, the known carbonate. Where's the missing carbonate on Mars? There's a lot of carbonate supposed to be there that no one's finding. All of those can be explained by one process, I think. So chlorocarbon-containing crystallites embedded are likely to be chlorocarbon clathrates, probably type 2, although phosgene is still a possibility. So is it a chloromethane clathrate hydrate, and does that explain the curiosity results? Well, I'm closer than anyone else. Let's leave it at that. electrochemical energy can drive organic synthesis on Mars and would do elsewhere. Europa, early Earth, modern Earth, Enceladus, and answers this serpentinization dichotomy, the fact that in experiments you can't make enough hydrogen to make methane in this system. You have to use magnetite in there. There's a Mayhew paper. And I've talked to like Alexis Templeton and Tom McCollum and all the guys that are premier in this field, and they all agree that the hydrogen could come from electrolysis of water, basically. And that's been a missing step for a while. So was this early Earth? If I'm right, and this process is widespread, what colour's Mars? It's red. Why is it red? It's because it's rusty, right? A lot of rusting has gone on Mars, and this process is linked to rusting. So where did the massive carbonate bodies go? Well, maybe there is carboxyl or carboxylate or oxalate. Who knows? But if that system is right, then you take away the need for hydrothermal energy and you transport it to electrochemical energy undertaking those reactions. And if that's correct, the whole seafloor where it contacts brine is now your basis for reaction for abiotic processes leading to prebiotic processes. That's the implication. It's arm wavy, but it is the implication, right? So we're moving away from these small hotspots or spreading centers to the entire surface area of where igneous provinces react with seawater. Maybe. Okay. So that has major implications for carbon cycling on Earth. Some of the stuff I'm doing is try to measure that in the lab. I'm going back and relearning electrochemistry. Not that I learned it very well in the first place, but that's what I'm going to do. And I'm going to throw this out as the ultimate arm-wavy end to this talk, all right? The Miller-Urey reaction that won the Nobel Prize in the 50s, people often joke that they advanced abiotic chemistry by 20 years and then spent 20 years holding it back. Because one of the things in this system is this doesn't make sense. It's obvious from context data that the early atmosphere of the Earth did not contain these reducing species. So the Miller-Urey reaction, which produces lovely amino acids, abiotic amino acid signals, and all of that, in this system was a gaseous, so these are fed in as gases, with a spark discharge that was lightning through here, and the condensate was all these amino acids in the aqueous phase down here. And basically, what I'm saying is that these are all produced and reacting together in the aqueous phase, and that the spark discharge is from electrochemical energy. Miller-Urey could have been right, just out of context. And that's where I'll leave it. Thank you very much. Yeah. So I have 4 d-spacing 3 indexes all line up for type 1 or type 2 clathrate. But they also line up for some of the phosgene, and that's the problem. It's not conclusive for any of those things. That's why I'm very careful to say a chlorocarbon-containing clathrate. But at that point in time, you know, on a 10, 15, 20 nanometer crystal, there's not much else I can do. But it's reproducible across all the crystallites that we analyzed. And the fact that it's, when I put a Raman laser on it, heat it a little bit, it just disappears. It suggests that it's not things like, say, a perchlorate, calcium perchlorate, or anything like that. It just doesn't index with any chlorine-containing mineral apart from the clathrates and phosgene, which chloromethane will degrade to phosgene with a bit of UV. So it could, even if it's chloromethane, it could be producing phosgene by oxidation.
I have a comment on that too.
Yeah.
So, you have a 17
Yeah. We got tilting information in 3 different planes as part of that. One way or another. Actually, we have the tilting data. We just haven't crunched it into that table. So thank you for that. That'll do it. Brilliant. Uh-oh. Yeah, they've been found in NACLA. It's inconclusive whether they've been, from what I know, it's inconclusive as to whether it's been found by Chemin, but there's a lot of allusion to possible halide being found in the APXS data. Elementally, it doesn't match, but we haven't. I don't think we've seen any halide from the Chemin observations that I can remember. But the problem with the Chemin XRD observations is it can be up to 35% amorphous material, so we're not actually getting good 100% signals from it. Which, you know, if you look at the stuff I was showing, is that it's all amorphous, coming from those corrosion processes. And then some of the stuff in the cracks and that lot is completely tiny grains, like 2-3 nanometer crystallites. Sometimes I've found siderite in my last look at that, but they're tiny. They're so small that I doubt Chemin would be able to actually index those.
So going back to the 2012 paper, correctly, the carbon had associated
Yeah.
electrochemical carbon.
Yeah.
Does that suggest you have a second generation carbon that is recycled the primordial carbon and associate that with the biological process?
Well, the thing I'm doing at the moment is going back to those spinels that we said were igneously formed. And so the resolution of my Raman single pixel XY is about 370 nanometers per pixel. But the cracks I'm looking at around these features are 20 nanometers in diameter. So I'm starting to wonder if I'm not looking at the same thing again, and that water or brine hasn't got through these tiny cracks and reacted with the magnetite again to do the same thing. So I'm questioning whether or not this is igneously produced anymore, and whether it's all the same process that I'm looking at. But it doesn't have to be. It could be a second generation. I produced a review paper last year showing 5 different synthesis mechanisms on Mars. Primary hydrothermal, like late stage, that's what we were calling the Tessint observations at the time. Late stage magmatic, there's still volatiles pouring out as the crystals have developed, and then they react as the temperature lowers. Secondary hydrothermal, like Allen Hills 8.4, which is obviously a secondary process dumping those carbonates. Igneously formed, the process that we talked about then. Then there's impact induced, or impact included, as in as an impactor has hit and carbon has come off that impactor, it's solidified in the meteorites. And impact induced, where some of the siderite you see in Allen Hills and elsewhere, if you heat siderite up above 450, you'll start to produce polyaromatic hydrocarbons and magnetite. And I think there's evidence for that in Allen Hills. So I think I have 5, up until here, I had 5 mechanisms. Now I'm going back and trying to disprove and connect those mechanisms together to see if I've not got overlap on those. Well, it's a true story, isn't it? Yeah, the nano. Yeah. Well, if it's true, I mean, I think it's exciting. As George can tell you, there's been a real debate in the early life field as to what the C1 compound was. people assume that carbon dioxide is too oxidizing to undertake these organic reactions, but in the system, if you're pumping protons into it and then functionalizing with chlorine, who knows? And that's roughly where I am with it all at the moment. Any other questions? Thank you very much.
Thank you very much, Steele.
Thank you.
Don't forget the mineralogy.
It's the most important thing.